Hi, this is a brief lecture on lipid metabolism, and the outline of the lecture is as the following. Right, the lipid metabolism starts with lipid ingestion, meaning the lipids, the triglyceride that you consume in the diet. So during the digestion process, this lipid will be emulsified by the bile salts and will be broken into smaller chain by the action of the pancreatic lipase and finally yielding free fatty acids. Alright, so this free fatty acid inside the lumen of the small intestine will be then packed into a structure called chylomicron. And basically, chylomicron is a vesicle which is made of triglyceride, cholesterol, and free fatty acid. This chylomicron will cross the intestinal membrane via a process called exocytosis, and then it will enter the lymphatic system, and following that it will be transported to the circulatory system, and from the circulatory system it will be either transported to the liver or it will be stored in fat cells as adipocytes. From the earlier slide, we have learned that the fatty acid is eventually transported to liver or stored in adipocyte as energy storage. In order for us to obtain energy from this fat, the triglycerides or the fats must first be broken down. This process is called lipolysis or beta oxidation. So in the first step, when the triglycerides go through a process called hydrolysis, it produces two principal components, which are fatty acids and glycerol. So this is the fatty acid you see right here. The fatty acid then will be converted into a molecule called fatty acyl-CoA. This reaction takes place in cytoplasm of the cell. Now, in order for us to generate energy, the fatty acyl-CoA must enter into the mitochondria. Remember that the mitochondria is the energy powerhouse of the cell, and this is where energy or ATP are being generated. And also mitochondria is an organelle with bimembrane. So the fatty acyl CoA could not pass through the membrane and enter the mitochondria. At this point, there is a transport system to transport the fatty acyl CoA into the mitochondria. This transport mechanism is known as carnitine shuttle. So during this reaction, the acyl CoA is transiently attached to the hydroxyl group of carnitine to form a molecule called fatty acyl carnitine. So this molecule then enters the mitochondria, and in mitochondria it will be converted back into fatty acyl CoA. Then this fatty acyl CoA will be converted into acetyl CoA. Then finally the acetyl CoA will enter the Krebs or the TCA cycle to produce ATP. Right, earlier we have looked at the process of beta oxidation or lipolysis or breakdown of fatty acids to yield energy. So what happens when there is abundance of high energy molecule such as ATP and NADH? So in this situation, the acetyl-CoA from the citric cycle or the TCA cycle will be diverted towards fatty acid synthesis. So you get your acetyl-CoA here. And remember that the CoA moiety could not transfer or pass through the membranes of mitochondria. So in order for it to cross the mitochondrial membranes, the acetyl-CoA needs a transport system. And here it uses the citrate shuttle. Alright, in the citrate shuttle, what happens is the acetyl-CoA will be combined with oxaloacetate, another molecule, to form citrate. Alright, this citrate then will cross the membrane and make its way into the cytoplasm. In cytoplasm, the citrate will be converted back into acetyl-CoA and oxaloacetate. The oxaloacetate then will be converted into malate and then into pyruvate, right? And then the pyruvate will actually go back into the mitochondria, all right, and wait for the next cycle of lipogenesis, where else the acetyl-CoA will be converted to malonyl-CoA and goes through a series of processes to be synthesized into fatty acids. So, and this is what happens in biosynthesis of fatty acid. So here you are looking at animation depicting fat tissues. So you can see the scanning electron micrograph of samples of fat tissue showing the fat cells, alright, the adipocytes in red and yellow, and you can see they are being surrounded by fine strands of the connective tissues. Earlier we have learned that the excessive fats will be stored as triglyceride in form of adipocytes. This is a long-term energy storage, and it's important when the body goes through starvation or a prolonged fasting. During this period, glucose is not available to the body, so the body needs an alternative energy source. Alright, the fatty acids, which comes from the breakdown of triglyceride, will go through series of reaction to produce this component called ketone bodies. And ketone bodies are important because they can be used as a source of energy for the brain. The brain normally just uses glucose, but in absence of glucose, ketone bodies can supply energy to the brain. So when ketone bodies are produced, they are being produced faster than they can be used, and the ketone bodies can be broken down into carbon dioxide and acetone. So acetone is being removed via exhalation, all right? And one of the symptoms when there is excessive production of ketone body or ketogenesis is that the patient's breath smells like alcohol due to the presence of acetone. All right, and this is actually clinically important because this is quite common among diabetic patients. All right, and ketone bodies are acidic in nature. So if there is excessive formation of ketone bodies, the patient will go through a condition called diabetic ketoacidosis, and it is a very dangerous condition. In this slide, you are looking at a series of reactions which the ketone body goes through in order for it to produce acetyl-CoA, which finally will be diverted to the Krebs cycle to produce energy. There are three main types of ketone bodies, which is beta hydroxybutyrate, acetoacetate, and also acetone. So here you can see the beta hydroxybutyrate is converted into acetoacetate and then followed by production of acetoacetyl-CoA. So at this stage, the molecule will be split into two, forming 2-acetyl-CoA, which then enters the Krebs cycle for formation or production of energy. Right, that's all for this mini lecture, and here I have provided the references. Thank you.